This is the MC1R protein. It is found in the plasma membrane of a melanocyte, and melanocytes are usually responsible for producing pigmentation in skin, hair, and eyes. Today, I'm specifically looking at fur color in mice, and it was found that beach mice have a lighter fur color while forest mice have a darker fur color. And that is due to a mutation that I will explain now. So I use clay to build this protein. All of the parts of the protein here are seen made out of clay, including the arginine, the MSH, adenyl cyclase, ATP camp, and then the rest is just whiteboard markers. So how the normal protein functions is that alpha MSH would bind to the MC1R protein at this arginine site at the 67th position. Then signal transduction would activate adenyl cyclase to produce CAMP from ATP, which then turned on the expression of TERP1 and TERP2 and P genes to then upregulate eumelanin. So this is the primary structure of the MC1R protein. This would just be constituted by a single chain of polypeptides all in a row. And specifically at number 67th position, you have an arginine. And an arginine would look like this. So arginine has this guanidino side chain group, and it is highly polar and hydrophilic. It has the positive charge, which makes it basic. It's larger compared to other amino acids because of the bulky guanidino group, and then because of this, it's able to hydrogen bond with other molecules. Next, we have the secondary structure of the protein, which is constituted by alpha helices and beta sheets. So this would be an alpha helix here with the helical form, and then a beta sheet would just be pleated with the proline causing the kinks. And next would be the tertiary structure, which would have side chain interactions causing salt bridges. There would also be hydrophobic interactions. There could be disulfide linkages and just more hydrogen bonding. And so the protein would just bond with itself essentially. And so here we have something different. This would be the point mutation. At the 67th position, the arginine has now changed to a cysteine. So now I will go ahead and talk about why cysteine causes this tertiary structure to not have hydrogen bonding going on anymore, not as much side chain interactions within the whole protein. And so cysteine has a side chain with a thiol group. It is uncharged, it's polar. It's much smaller than arginine is going to be, but it does have this ability to form disulfide bonds. And just to see that on a larger scale, now cysteine is here instead of the arginine on the loop of the protein. And so alpha MSH will not be able to bind to this cysteine because it has a different side chain. It can't interact anymore. Therefore, like I said before, there will be no signal transduction down. Adenyl cyclase cannot convert ATP into CAMP, and then therefore there will be less expression of TERP1, TERP2, and P genes, and a down regulation of eumelanin. And eumelanin causes darker fur. So if it down regulates, then you're going to have the lighter fur color, and this is the exact mutation that causes the lighter fur color in mice. And so just to wrap things up, some limitations of this model, you can't see individual protein residues right here, but that would constitute the primary structure of the protein. Also, there are lots of other cellular processes that are surrounding the specific gene, and so this wouldn't be so prominent. There would be lots of other things going on all at the same time, and it'd be moving very quickly, not stationary. And lastly, there is an actual conformational change of the loop that can't be seen in this model that the cysteine charge has. So thank you.